All right, so we're talking about the cell cycle today and how our bodies need to generate new cells, all right? Generally speaking, there are two cell types in our bodies, right? We have sex cells, which are utilized for reproduction, and then we have somatic cells, right? These somatic cells are what we sometimes call our body cells, right? They are the cells that gives our body its function to make tissues and so forth. So we have to continuously make more of these cells. And so there are two processes that happen biologically to generate new cells, right? So the cell cycle is a very important principle that we need to sort of cover as we look at these characteristics of life and how we continue to exist. Our bodies need to make new cells. Let's see, I see something in the chat. Okay, I'll get your question in just a second, David. So we're going to look at what happens with the eukaryotic cell cycle. All right, I have a few slides that I'm going to go through today, and then you're going to embark a little bit more independently with an interactive exercise and some follow-up questions. So let's go. The eukaryotic cell cycle. So this is true in eukaryotes, right? That includes us, right? So as I said before, like our cells do not just live infinitely, right? They're going to reach a certain point where they're either going to stop growing or they're going to start to divide, right? To produce new cells, right? And so this process of cell division, right? Our body's making new cells is a highly regulated process, okay? The process or time that it takes for a eukaryotic cell to complete a cycle is referred to as generation time. Write down that term, generation time, right? Generation time is the time that it takes to complete an entire cycle, all right? So we're gonna take a look at this cycle. What happens during the eukaryotic cell cycle, all right? And we'll get a little bit more details about this in our lecture course, but for right now in lab, we want you to just take a general look at what's happening during the eukaryotic cell cycle. So I have a schematic here that you're looking at, all right? The eukaryotic cell cycle can be broken into two general phases, okay? Write down the phase, the term interphase, right? Interphase is written right here. This is a collection of activities that's happening before the cell actually gets ready to divide, all right? Let me back up a minute here before we go through this cycle. So we're establishing that new cells have to be made, right? These new cells are generated from pre-existing cells, all right? So some things need to happen for this new daughter cell to be created. And the steps that need to occur before a new cell is generated, it's termed the cell cycle, okay? We're gonna go through the steps that are necessary for a new cell to be generated. Okay, the eukaryotic cell cycle is characterized by interphase, right? There are some things going on during interphase, and then there is a phase known as mitosis, all right? Interphase and mitosis. So what's happening during interphase? There are three steps that occur during interphase, all right? So generally speaking, in your notes, what you're first gonna highlight is that interphase is like a preparation period. Growth, yes, G1, S, and G2, all right? 3 steps of interphase. And this is our preparation period. This is when our cells are growing, becoming happy, metabolizing enzymes and other factors that are needed for mitosis to take place. So it's a collective preparation period. This is interphase, all right? There are 3 stages in interphase that you need to write down: G1, S, and G2. All right. So we're saying here that the eukaryotic cell cycle, it has interphase, and then it has mitosis, which is a whole nother set of steps that are taking place in preparation for division. Okay. Mitosis is going to produce an exact copy of the parent cell in this daughter cell. All right. And so we're going to run through what's happening with mitosis as well today. All right. So let's go back to interphase. All right. The first part of the cell cycle is interphase. The cell spends most of its time in interphase because a lot here is happening. At G1, again, we're at a checkpoint phase. We're making sure that this cell is growing. It's nice and happy here at G1, right? Now, we're getting ready to divide. The ultimate goal here is to divide, to go from one cell into two cells, right? And during mitosis, we're creating a complete replica of that parent cell. So however many chromosomes or whatever DNA content exists in the parent cell has to be made and available for the daughter cell. So there's a period of DNA synthesis that takes place, okay? So for humans, we have how many chromosomes in our bodies? How many chromosomes do we have in total?
23.
Say it again.
46.
46, so 23 pairs, right? So however many chromosomes are in this cell, during the DNA synthesis phase, as we're getting ready to make a new cell, what happens during DNA synthesis with our chromosomes? Does anybody remember? Yeah, I think what are you saying? Well, it doesn't split yet, but before it splits, what needs to happen? If our daughter cell is going to be created to be an exact replica of the parent cell, what needs to happen with the DNA in the parent cell? It has to duplicate. Yes, right? So G1 is a gap phase where, again, kind of a checkpoint, making sure enzymes and things are in place. And then we go into synthesis, right? This synthesis phase is critical. It's absolutely critical. During this S phase, right, we're right here in the cell cycle. During synthesis, our DNA is being duplicated or replicated, right? It has to be doubled because at the end of mitosis, the process of cytokinesis is when this parent cell is going to split in half to generate a new cell. And we want this new cell to have the exact same number of chromosomes as the parent cell started with, right? So we got G phase, we've got S phase, and then we got G1, and we've got S, and then we've got G2. What's happening at G2? This is another checkpoint. This is kind of our final checkpoint now before we go over into mitosis, okay? This G2 checkpoint is making sure that replication has happened the way that it's supposed to, that there are no errors. It's checking for fidelity because, again, we don't want to pass off any abnormalities into that daughter cell, right? So that second G2 checkpoint is critical. Absolutely. So there are mechanisms in place to detect abnormalities and when things have not happened the way that they should, and those cells are signaled to die. They will not continue through the cell cycle. Absolutely. Right. So this cell cycle process, I want you to conceptualize this. It is it's a highly regulated process, right? There are checkpoints and mechanisms in place that if things haven't happened the way that they should, these cells are marked to die. All right. So interphase, what's happening during interphase? Is it a period of preparation, a period of DNA synthesis? Everything is happening in interphase. The cell spends most of its time in interphase, but what's not happening in interphase is actual cell division. Okay, the number of cells do not change during interphase. Okay, so the second part of the cell cycle is mitosis. Okay, mitotic cell division, again, write this down in your notes. The end result are identical copies of the parent cell. So we're generating new daughter cells that are going to be replicas. So mitotic cell division is sometimes thought of like cloning. We're making exact copies. Okay, so what's happening during mitosis? Now, mitosis is a little bit more involved. All right, we're going to walk through the phases of mitosis, which is still kind of a preparation for division. Okay, the actual division of the cytoplasm happens during a phase known as cytokinesis. Okay, write this term down right here. When we get through with mitosis, we're going to see that the final stage in this cell cycle, right, as we're generating this new daughter cell, that process is called cytokinesis. Okay, when that parent cell actually splits from one cell to two. Okay, so we're establishing right now the steps that need to happen. So interphase happens. We've got G1, S, and G2 before we actually move into mitosis. All right, let's walk through what's happening during what questions do we have about interphase? All right, so I do want you to know all the phases of the cell cycle. All right, so interphase is the first half, right, where we have G1, S, and G2. All right, checkpoints, DNA replication, making sure everything has happened the way that it should with fidelity. If it passes G2 and goes on into mitosis, and then we're ready to create a new daughter cell. So there are 4 stages of mitosis. Write it down, PMAT, 4 stages of mitosis. Mitosis is characterized by prophase, metaphase, anaphase, and telophase. This should not be anything that you haven't heard before. Okay, PMAT. PMAT, what is happening during mitosis? Mitosis is, again, we're getting ready to split this parent cell into a daughter cell. So some things need to happen as far as this chromosome alignment, right? And we're clear here, we're talking about these chromosomes. Our DNA, our genetic material is the point of focus here during the cell cycle. Okay, when we're generating these new cells that we're making have to be an exact copy of the parent cell. All right, so what's happening during prophase? A lot's happening, but most importantly, what we're seeing is that these chromosomes start to condense and become visible. All right, during metaphase, we see that these chromosomes are now starting to align towards the center of the cell. All right, anaphase, now we got these sister chromatids starting to separate and move towards opposite ends of the cell, right? Telophase, now we're at we have 2 different sets of chromosomes or chromatids. We have 2 a distinct sort of nuclear envelopes forming at each end of the cell, because the end goal here is that this parent cell that we started with is going to now end up being divided into 2 new cells, right? So we're going through the steps of preparing these chromatids, these sister chromatids, these chromosomes and sister chromatids, in order to form 2 separate regions of chromosome alignment in order for these 2 new cells to be created during cytokinesis. All right, so real quick, prophase, metaphase, anaphase, telophase. This is PMAT. This is what happens during mitosis. This is this cell now preparing to divide and make a copy of itself. And the copy that we end up with needs to be a direct replica of the cell that we started with. All right. So what's happening during prophase? As I said before, what I want you to know simply is that these chromosomes are starting to condense. A chromatin, which is DNA and protein assemblies, are starting to condense and become visible chromosomes. All right. This is our chromosome. All right. During prophase, they are starting to condense and become visible. All right. You see these sister chromatids coming together in a pair. All right. So prophase, we've got chromatin condensing to form chromosomes. These chromosomes are becoming visible. All right. This is the first step of mitosis. All right. Then we have metaphase. All right. Metaphase, again, is the second step of mitosis, where we start to see these chromosomes line up along the midplate or center of the cell. Okay, very obvious midplate alignment. Okay, the way I remember metaphase is M, meta for middle. All right, so if I showed you some pictures and images and asked you to identify the various phases of mitosis, I expect you to be able to do that for me. Okay. During metaphase, we've got these chromosomes are highly condensed. They formed into chromosomes that we can easily see. They're aligning towards the middle of the cell. Okay, that's what's happening during metaphase. Anaphase. Now, again, remember our end goal here is that we're trying to end up with 2 distinct cells that have the same chromosome content. So that's exactly what's happening. These sister chromatids are now going to start to separate and move towards opposite ends of the pole. And that's what we see right here, right? They're going to start to separate. All right, so each of these sister chromatids are going to be single chromosomes now that are going to be needed to, again, create these new daughter cells with the same chromosomal content. All right, so anaphase, we got these sister chromatids separating. They're moving towards opposite ends of the pole. This is a very clear schematic of what's happening. If I showed you a picture of this, I would expect that you can identify this as anaphase. All right, the final step of mitosis is now we have two distinct ends of the cell, right? Where these chromosomes have sort of decondensed and they're ready to basically exist as their own nuclear capture of chromosomes, right? Each of these are going to have the identical genetic content as each of these new sets of chromatins are going to have the same genetic content as the parent cell. All right, so we've got that this cell has enlarged, right? These chromatids have moved to opposite ends of the pole. We're going to start to see sort of a nuclear envelope forming around each of these distinct groups of chromosomes, right? You're going to start to see some indentation in the cell membrane, right? And a process known as cytokinesis is now getting ready to occur, right? Cytokinesis is the final step where this cytoplasm now will divide into two daughter cells, okay? So this happens right after telophase, right? So we've gone through prophase where the chromosomes condense. Metaphase, they start to align up along the mid plate of the cell. Anaphase, you see these sister chromatids now start to separate and move towards opposite ends of the poles. And then during telophase, we've got two distinct groups of chromosome now where you start to see this nuclear envelope forming around these two distinct groups of chromosomes on opposite sides. And now this cell is ready to divide. Okay. Cytokinesis is the actual process of this cell splitting from one into two. All right, this little indentation right here, I do want you to know, I mean, I know we're going over some brief details, but I do want you to know that this little indentation that starts to form in the cytoplasm before cytokinesis happens is called a cleavage furrow. All right, we see this cleavage furrow in animal cells, right, as it's preparing to split.
Oh, yeah, but yeah, I can't say that for you, though.
Can you mute your mic, please? All right, so again, what we're seeing here is before this cell actually splits off, you're going to see this sort of indentation happening. All right, where we're getting ready to get two identical daughter cells. All right, I'm going to stop right here because you have some things that you need to do on your own. But for the purpose of today's lesson, you need to understand, why is the cell cycle essential? How does it take place? Okay, what's happening during the eukaryotic cell cycle? We've got interphase, which is characterized by G1, S, and G2. G1 is gap phase 1, where it's a period of preparation where the cell is growing and obtaining nutrients and getting all the things that it needs. S phase is the synthesis phase, where the DNA is being replicated or duplicated. G2 is another gap phase 2, where it's another checkpoint that before this cell goes into mitosis, we need to make sure that everything has happened the way that it should. Once it gets into mitosis, these chromosomes are going to start to have more activity. Okay. M phase mitosis is characterized by PMAT. Right? What does PMAT stand for? Prophase, Metaphase,
Anaphase,
Anaphase, and Telophase. Okay. You need to know what's happening during PMAT before the cell actually gets to cytokinesis. All right. The end result of mitosis, write this down, are two identical daughter cells. Identical. So sometimes we think about mitosis as cloning. We're making an exact replica of the cell that we started with. Okay, we talked about the cell theory in our lecture class, and we know that one of the postulates of the cell theory is that new cells arise from pre-existing cells. Okay, we see this in the cell cycle. Please mute your mic.
Mm-hmm. Mm-hmm.
Okay. All right, so we're going to stop here today. If you log into your Blackboard course, there is a title under your content area that says cell cycle. Okay, there's a link for an interactive exercise that you're going to work through. You're going to annotate in your notes the different phases of the cell cycle that we just talked about briefly. You're going to work through those animations. You know, click on each hyperlink. It's going to show you what's happening during each phase. Click on all of the hyperlinks, and then there is a exercise that you're going to turn in. Okay, you do have the rest of the day to get this done. Okay, you